Hey, everybody. Welcome to yet another Journal Club brought to you by Lifespan.io. I'm Alden Levitek, as per usual. And we have yet another wonderful journal article brought to you by our organization on the topic of longevity and aging. So we're going to be focusing on a specific organ system for this journal club. And welcome to everybody here who's participating. And we're going to be focusing on skin aging in this journal club. We've done a number of different journal clubs looking at all sorts of organ systems, the brain, I believe, kidney function, lungs, blood. I don't think we've done skin yet, Steve. I think skin is one of the major organs that we haven't actually. Maybe a little bit.
I think we did. I'm going to have to countermand that. I'm pretty sure we did one a few years ago about skin rejuvenation, and I can't remember what the method was. I really don't have an eidetic memory, so unless Keith was with us, he would remember. But I definitely know we've done something to do with skin. I think. Yeah. Oh, I know what it was. There was a cream that alleged to be able to reverse signs of aging, but they used like an epigenetic approach. But that was about 4 years ago, so I'd forgive you for not remembering.
Well, it's amazing that you remember that. That was about 4 years ago. But I think in this paper here, they really drill down deep and they look at a particular factor. So that was a topical cream that had some sort of effect, and I can't remember what the epigenetic modifications were. And I'm not sure if they actually drill down into a pathway. But this paper here, they really go into a pathway, and this is from the Technion Institute. So let me pull up this paper here, share my screen. Whoops, where's my, close this here. There we go. Expand this. Close this. Oops.
Oh dear, Oliver seems to have disappeared again. Oh, here he goes. He's back again, guys. I was going to start talking about Star Wars, but now he's back. Whew, you've dodged a bullet there. You don't want to see a middle-aged guy ranting about Star Wars. You just don't. Here he is.
And I'm back. Slightly unstable connection, but here we are. So let's go to the paper. Share my screen. Try that again. Sometimes we get a glitch here in the matrix. Why is that share button not working? Zoom is having some weirdness that I haven't experienced before here. Bear with me for one moment. Why is my share feature not working? I think I can share. Should be able to share. I did share just a few minutes ago. That is weird. There we go. That should be visible to all. All right. Human Organ Rejuvenation by VEGF-A, Lessons from the Skin. Kind of reads almost like a review paper, isn't it? Aviat Karen et al. From the Technion Institute. And so what is human organ rejuvenation? So we'll discuss that in a moment. So what is VEGF-A? So that stands for vascular, I believe, endothelial growth factor A. So this is a paper where they basically identify this factor as both necessary and maybe a little bit more importantly, sufficient for the rejuvenation of skin. And they have a kind of a unique model here. So should we admit Nora Gideon into the waiting room? Or am I in charge of that? No? Okay. So what did they do in this paper? So they have a unique mouse model here where they basically use mice that are immunocompromised, so highly immunocompromised. And this work actually goes back from these authors quite a bit. There's some of the authors have done work identifying basically using this model system to look at skin aging, going back to, I believe, almost 30 years. There's papers going back from the 90s. And in this paper here, they really focus in on identifying one of the key factors, VEGF-A, which is basically a polypeptide, a protein, and it docks to a receptor found on endothelial cells. So they have a mouse model here, which is, again, kind of another unique contribution. So they use these mice that are referred to as SCID mice. And that basically stands for severe combined immunodeficiency mice. So these mice are very immunocompromised, meaning you can transplant all sorts of stuff into these mice and they won't reject it. So normally when you try to transplant tissues from one organism to another, you have to have a very close match. So the tissues have to match. There's proteins, major histocompatibility complex proteins that basically will recognize self versus non-self. So this is, a cornerstone of transplantation biology. And if you don't have a perfect match, you have to take drugs that suppress the immune system, et cetera, et cetera. So you can't just transplant tissues from one individual, one species to another unless they're really closely matched. So if you're going to do this in mice or humans, they have to be really, really, really matched, like twins, for example, or somebody very, very closely related. Or if it's in mice that are kind of bred to be almost clonal, practically, right? So their systems are very similar. These mice are so defective and immunocompromised that you can actually take tissues from not just any old mice and transplant it to this mouse, but you can take tissues from other species like humans and basically stick it into the mouse organ system, and that tissue will survive as long as it's obtains the nutrients and is vascularized. That tissue can be transplanted into the mice, and these mice have been used in a number of different studies for many years. And here they're applying this to the study of aged skin. So this sort of really busy figure here is an overview of the experimental designs that they've done in this paper, where in figure 1A, you sort of have a kind of the beginning of the project, where you take biopsy samples from young individuals and old individuals, and then transplant them into either young or old mice, right? And let me see if I can recall how old the humans were. I have to go into the materials and methods on this. Everybody here read this paper, right? So if anybody wants to chime in and tell me how old the person was, it's not critical. I'm going to assume that it's somebody who's over the age of 60. It'll be in the materials and methods, so we can pull that up.
43 and 83.
43 and 83. Oh, I would have expected maybe a little bit younger, but that's fine. 43 is young enough. 43 is younger than me, so that's young. So they've taken skin biopsies from young and transplanted them into the epidermis, into, well, not just the epidermis, but the dermis as well, tissue plug, into young mice and old mice. And then they have these acronyms for these transplantations. So young into young, meaning young into young mice, young human into young mice, young into old, right? So that's young participants into old mice, and then old into young and old into old. So these are your controls here. And then they did studies to see what happens. And these rejuvenation experiments, a lot of them, weeks post-transplant were 4 weeks. So about a month after transplantation, they did a lot of these analyses, right? So rejuvenation experiments, loss of function experiments, and gain of function experiments. So loss of function, looking at what happens when they block VEGF or if they add VEGF. We'll look at that later. And rejuvenation experiments, meaning taking a look and seeing what happens to the tissue, and looking at a wide variety of parameters. And these are some of the parameters they looked at, which is going to be unpacked in the next few figures. So here's just a kind of an overview, right? So you've got these tissue sections placed into mice, and a variety of different experiments have been done, and a lot of factors have been looked at. So we'll look at some of these factors that correlate with aging skin, right? So levels of SIRT1, which is we've talked about in the past, which is a histone deacetylase required for well, a lot of things, but let's say youthfulness, collagen production, so on and so forth, and the expression of a whole bunch of transcripts that correlate with aging or rejuvenated tissue. And what they've done also, very interestingly, if I can pull up the supplemental, it's not actually in these figures, it's in the supplemental. They actually looked at aged skin that they've then added into tissue culture. So this is not in mice, this is in tissue culture. So in a petri dish that's supplemented with growth media, but without any other serum or factors that can kind of confound the results, and kept alive. And then treating it with just VEGF-A. So some of these experiments you'll see, and this is, to me, this is sort of the kind of one critical part of this experiment, is to show that VEGF is actually not just required, but is necessary in of itself, right? So just having VEGF-A is the important component of this. And we'll see why that matters, because VEGF-A is vascular endothelial growth factor, and it promotes blood vessel growth. That's kind of its main thing. So if you add any of these skin sections into a mouse, it's going to promote, you'll see blood vessels basically developing, which is dependent on the presence of expression of VEGF-A. And if you have blood vessels forming, well, you have a lot of things getting into this tissue from the mouse. So it doesn't discount the possibility that other factors are involved in the rejuvenation of the long-term rejuvenation of the skin sample because of the presence of blood vessels. But VEGF-A we'll see, is one of the key components. Okay, so I'm just going to stop sharing here for a moment. Anybody has questions about just sort of the broad overview of what I went over, feel free to ask. Silence.
We're just astounded by your excellent explanation. So,
or just confusing. All right. So, well, one thing to keep in mind about VEGF-A, it's important in tissue rejuvenation, but it is, everything in biology, everything in, human systems, mammalian systems is pretty complicated. Things have, it's a yin and yang. There's, too much of a good thing, right? Everything has to be regulated to a proper dosage. So before everybody goes out there and starts getting purified VEGF-A, I'm not sure how expensive that is, probably ridiculously expensive, and giving themselves an IV drip, just realize that it's also overexpressed in tumors, right? Because tumors, angiogenesis is a key component of tumors. So having too much blood vessel growth is also a bad thing, right? So that's just one caveat there, right? So if you're wondering, how critical these dosages are, the authors state that later in the paper, that dosing regimens will probably be a critical component of this. Okay, let's go back to sharing our screen. Okay, so one thing that they look at is basically how the tissue performs after transplantation. One critical component of this experiment is obviously the negative control of going not just old into young, but old into old mice. And these are cross sections of mice. These are stained. So hematoxylin and eosin staining is kind of a traditional stain that's used in histology to basically stain acidic and basic components, meaning DNA and proteins, and things take on this pink and purple stain. So these are like classic histological stains where you could see, kind of main parameters of how tissues look, right? So here's young skin, and these acronyms basically. So I think SC is subcutaneous, right? And you see these involutions that I believe they're pronounced RETES or RETES. So young skin has this kind of convoluted structure, sort of like the villi you would see in an intestine, right? So, and this is high surface area between the epidermis and the dermis basically, performs a wide variety of functions, but one of them is that it basically helps to tether the skin, right? Makes it more attached, if you will, kind of to the basal layer. And old skin kind of has this flattened characteristic, as you see here, right? So this thickness is one of the components that they are going to be looking at. So I'm getting calls in the chat that my screen is not visible. Is that visible? Is it okay? Can you see it? It is visible.
I can see it.
Okay. So sorry, Sven. Maybe. Okay. So maybe there's a random Zoom problem all over the world here, where things are just kind of getting weird with Zoom. Anyway, so one thing to keep in mind is one thing that the authors mention is that these are transplantation experiments. It's not when you do experiments you have to have controls, because there could be other variables at play that lead to tissue rejuvenation that might not have anything to do with the young mouse milieu per se. It could just be the procedure itself, cutting into the mouse, doing the surgery, doing the transplantation. You generate a lot of tissue damage doing that, as careful as you can be. And just the procedure itself might release all sorts of growth factors and things that can stimulate tissue growth and lead to quote unquote rejuvenation. And that's got nothing to do with the young milieu, and it's got everything to do with the actual surgical process. It turns out that's not the case here, which is what the authors controlled for. So they looked at old into old, and you can see here with the thickness in microns. This is after 1 month. Before old into old, there's no significant thickness difference, but you get this reestablishment of these involutions, these RETES. Hope I'm pronouncing that correctly. First time I've heard that word, RETES. These involutions between the epidermis and the dermis, and it's quite significantly thicker after 1 month, right? So all these experiments, one caveat is that these experiments are, well, except for the latter ones where they actually do allow the mice to age. These here that we're looking at are 1 month duration, right? And when we get to the tissue culture experiments, they're actually much shorter duration. They're 6 days. So just keep that in mind. So they look at a number of other parameters. So these are sort of the high overview changes that happened before they pinpoint exactly what's happening at the molecular level. But the changes are all positive, right? At sort of the kind of the gross histological level, which is these, this change from a thinner skin to a thicker skin after going and transplanting into young mice after one month. So they look at a bunch of other factors that correlate with rejuvenated or more youthful skin. They look at the number of cells proliferating. Let's see what stain they use for the proliferation. I want to say it's a DNA stain, but I have to go into the materials and methods. There's a number of different types of cells that are found in tissues that in the dermis that perform a number of functions, such as melanocytes, which basically lead to skin pigmentation. They perform a number of other vital functions. And those numbers go down as tissues age, as skin ages, but they go back up after 1 month from old into young. P16, which is a tumor suppressor gene, which basically is found in senescent cells. So it's the inverse of proliferation. Cells are not proliferating. That goes up. I don't know why it goes up higher in old into old, but it is significantly repressed. It goes back down if it's really down when it's old into young.
Maybe it's because of healing processes.
It could be because of healing processes. Yeah, in this case, because of the in this case, like I mentioned earlier, and that you mentioned now, Oren, it could be because of the surgical procedure here. SIRT1 levels go up. Mitochondrial factor, MTCO1, which I believe is that required for mitochondrial encoded cytochrome C oxidase levels. Those are significantly upregulated. So you have a lot of these parameters that correlate with more a youthful state going up when you do this transplantation, both basically at sort of the gross histological level, and a lot of these factors that are being stained for here in these immunohistochemistry slides that correspond to a more youthful state. All right. So are there any questions?
I just want to note it would have been nicer here if we also saw the yang in yang. It's like a positive control.
Yeah. To see basically if there's any confounding effects from the procedure in a young mouse, right?
No, I mean even the transplantation of young skin to young mice, just to see how far it is from the old in young.
Yeah, I don't know if they have that I don't think they have that in the supplemental. So I think that is something that's left out here. Yeah. So that would have been nice to see. Okay, so let's take a look at figure 3. So biomarkers related to epidermal skin aging. So more biomarkers are being looked at. And again, so this is a correlation between, things that are getting back to a more youthful state. Collagen 17A. So this is a type of collagen. So collagen levels in general, especially these large molecular weight collagens, go down as we age. That goes up. Filaggrin is, I believe that's a protein that's involved in, I don't know if it's one of these adhesion proteins, but it's involved in basically maintaining the hydrophobic barrier, I believe, in skin, right? So basically making it more intact and durable, so less things can diffuse into your skin, right? So those levels of this protein go up. MMP1, matrix metalloprotein, that goes down. I do know that these MMP1 proteins are correlated with, cancers do upregulate them. I don't know exactly why they would go down or go up in aging in general, but that goes back into a more youthful state. And they use picrocereus red collagen stain to basically show that these thick filaments, which are involved, which go down with aging, go back up again, versus these thinner filaments, that number goes down. So that's an inverse correlation with this type of collagen. The one question I would have with the collagen experiments, and those are really interesting, and I know we've talked about this before, Steve. I think Steve's still there. Is I'm wondering if, I don't think they've done these experiments, but collagen gets, a lot of these proteins get cross-linked as we get older. So I'd be curious to see if, the thicker filament levels of collagen go up, filaggrin goes up, all these things that go down, go back up. But there's a lot of other biochemical changes that happen in skin, and cross-linking is one of them. And there's a lot of, hey, Steve. So there's a lot of companies that are looking at anti-cross-linking agents, right, to basically make skin more pliable because it gets stiffer when it gets cross-linked. So I would be curious to know if the cross-linking of collagen and other factors, other proteins, also goes down. I don't know. I don't think they've. That would be an experiment, like a follow-up experiment. I'd be interested in seeing, but all the other factors seem to be pretty positive. So, any questions? Skin is getting rejuvenated as we go through the figures here.
Yeah, when can I get this? I'm a little bit, just around here, a little bit.
I could probably talk to Red Miller, who's a protein purification expert at the Cambar Center at Cooper Union, and she might be able to. We might be able to get you a discount on VEGF-A protein.
What you really meant, Oliver, was don't try this at home, folks.
Yeah, well,
you did, didn't you?
Unlikely. If anybody's going to try this type of experimentation at home, they've got the lucre to do it, because this is not going to be easy or cheap, unless of course you can come up with a compound or an intervention which the authors mentioned that can upregulate VEGF-A levels without actually injecting VEGF-A, right? So indirectly boost levels somehow, and that might be interesting, looking for small compounds or some sort of compound you could administer topically that can boost VEGF-A levels. That would be probably the more useful approach.
NAD precursor springs to mind. I think there was some things done in that direction.
Yeah, so
hyaluronic acid is another thing that seems to improve wrinkles. There was a study recently that showed that, I think it was a 12-week period of taking it. Actually, they used AI to look at the skin samples rather than what they would have traditionally done, looking at photographs, which is somewhat subjective. And the AI revealed that the wrinkles in the participants was noticeably smoothed. So, I'm not saying that you should necessarily take it, but I'm just putting that out there. I'm not selling it, by the way. So I'm not married to any brand, but I do use. I'm a little bit vain, so I use hyaluronic acid.
All right,
it's worth a shot, right? Brad Stanfield says it seems to be well supported, so it's one of the few things I take.
So we'll see what all these. So yeah, that's a good series of follow-up experiments, is to see what a lot of these interventions do to VEGF-A levels, right? So that would be interesting to see if those levels actually go up, if you get an improvement in skin. So yeah, I don't think they've done any other types of skin kind of experiments here. I know that there's a traditional approach for skin elasticity that I think, now that you've jogged my memory, Steve, in that paper from 4 years ago, I think they did a skin elasticity measurement where you sort of pinch the skin with a certain calipers, and if your skin has high elasticity and your collagen levels are normal and they're not cross-linked, if you let go, it springs back, right? And there's a way you can measure basically how, quantify how much springiness there is, and that gives you one determinant of youthfulness, right, for skin. And I think that's what they did in that paper years ago.
Yeah, it's a nice simple biomarker. Skin of course is I've said this before, I think even at Journal Club, skin is a really, really good target for aging studies and proof of concepts, because one, it's highly accessible and it's easy to test. Two, it's noticeable, so any improvements are noticeable, so it's potentially a high impact thing. So if something does rejuvenate the skin, it's very different from, say, I don't know, your liver or your kidneys, which obviously we don't normally have on the outside unless there's been an unfortunate accident. So yeah, skin is a great candidate, and I think we should be focusing more as a field on rejuvenation of skin, because I think it's the one that's going to have the highest impact and get people, well, probably interested. Let's face it, most people are vain, and there's nothing wrong with that. There's nothing wrong with wanting to look good, right?
Yeah. And Sven just mentioned that one of the caveats with using that skin pinch test with calipers is that, of course, that also depends on other factors like hydration of the skin, right? So if you're dehydrated, that's going to have an effect. So, I would assume if you're going to do this type of analysis, or that you would make sure your participants are hydrated and a
large cohort ideally as well, Oliver, to reduce the influence of outliers. So, if there's 10 people and one person's very dehydrated, it's really going to skew your figures. But if you test 100 people or 1,000 people, then that's going to be less of a statistical anomaly. I'm just putting it out there. But you know this. It's what you do as a job. But maybe folks at home don't.
Yes, indeed. Many confounding variables that need to be controlled for. And we've got some controls here, and some controls that we'd like to see as well. So, okay, so hopefully you can see the screen. We're getting to our next figure, and this is where we get to what VEGF actually does, and they look at a number of factors that are upregulated using I believe QRT-PCR, which is basically looking at so figure F is looking at transcript levels, so RNA transcription, for example, levels, and this volcano plot, which we'll just take a look at in a moment here, is looking at, I believe these are gene expressions or transcript levels of a whole bunch of genes after so I believe this is post-transplantation of old into young. So they look at which factors get upregulated. So going into figure 4A, they look at a marker for blood vessels. That's this CD31 plus marker in old into young. So what is old into young? So old human into young mice compared with old into old. And what they're doing here is, I believe, so this is transplantation of human skin. So this is looking at a cross section that's within the human kind of, I guess, transplantation area. And they stain for blood vessels, and they show that amongst many other parameters that change, you get more vasculature happening, as you would expect, but even more so if you go from old into young. So you get more vascularization, and this vascularization is happening is derived from, and this is kind of critical, is derived from human tissues. So they stain for human CD31, which is the vascular marker, and mouse CD31. So what you're seeing here from old into old, there's no significance, and old into young. Well, there is some, I don't know if there's that much, I don't think there's a significant difference, but there's a significant increase in vascularization happening within the transplanted dermis of humans into the mouse, and that vascularization is actually happening through the human cells being stimulated by something from the mouse. So it's not that blood vessels are coming in from the mouse and vascularizing it, but rather the cells themselves within the human dermis are basically are undergoing angiogenesis. So basically forming blood vessels. And they look at a number of different things that change. So this is sort of where they go into looking as to which factors are responsible for all of these changes, or at least some of these changes. And there's a lot of things that are upregulated. And, so you have pro-inflammatory factors go down, you have factors required for angiogenesis go up. VEGF-A is buried in here. And interestingly, I don't think VEGF-A is like the biggest change. There's other factors like HIF-1A, which they talk about later in the paper, that seem to have more of an expression. But so if this is this volcano plot, which basically looks at things that are downregulated and upregulated, from the right to the left, these are sort of some of the major factors that are upregulated, VEGF-A being one of them. in these plots. So they identify one of these factors, and it's known that it's required for vascularization, and that is what's occurring. So they start to focus on VEGF-A and other factors, but VEGF-A being one of the key ones that they then start to look at.
Oliver?
Yeah.
I just want to note that maybe I'm nitpicking, but here, when you look at the volcano plot, you see many, many, many genes. So we don't know exactly how they choose VEGF-A. It seems to me like they just know other papers that say that VEGF-A is important. There was like a science paper from a year ago, very impressive one, like saying that this is another hallmark of aging. And it's also like I guess they know the scientists that wrote this paper. So that's a bit weird here.
That's a very good comment, and when you do look at these plots and you see a lot of these other factors that are undergo changes, they get upregulated, not just VEGF-A, and then the question becomes, well, how did you why focus so much on VEGF-A versus other factors? And a lot of this work is standing on other papers that these authors and other authors have published that kind of fill in the gap, so to speak. And VEGF-A was probably identified as a potential lead candidate in a number of other papers, and this sort of adds to the evidence that VEGF-A is playing a role. Clearly, some of the experiments that they do with VEGF-A, like knocking it down using antibodies as a control in later figures in this paper, they get applied to a lot of these others as well to see what their contributions are. And the authors kind of, I'm not going to say hand wave it a little bit, but they do mention that VEGF-A in latter experiments in this paper, they'll show that it's actually critical for all these changes that we've seen before to actually occur. Again, given the fact that that these experiments are being done within a month, the histology in the mice, and 6 days in tissue culture, a lot of these other factors might be just as critical for longer term rejuvenation. If we start to kind of try to maintain the tissue for longer periods of time. So that's not to say that VEGF-A is the only one and only factor. We've sort of heard that before with other factors, and a lot of these pathways are very, very, very intertwined. So certainly certain factors are important. They might be the ones that kind of initiate a cascade of changes, and the authors propose a kind of a model at the end of the paper, which we'll look at. But VEGF-A, of course, is dependent on crosstalk with a number of other components upstream and downstream. So it's you can't just have VEGF-A and nothing else, right? But it is a critical component that the authors point out. Okay, so zoom into Figure 5. So this is Figure 5 is a little bit different in that this is post-transplantation from old into young, but then they let these mice age, right? So they keep these mice actually around for 12 months. So the mice are now these young mice are now becoming old and older mice, and they show that that these biomarkers change. So this to maintain this tissue rejuvenation from old into young. It's dependent on the young tissue milieu. If the mice are allowed to kind of age, right, gracefully or whatever in their environment, in their cages, these effects that we saw earlier in the earlier figure, I believe Figure 2, they get reversed, right? So it's no longer this older tissue is no longer is no longer, this older mouse tissue is no longer able to maintain this rejuvenation effect. So SIRT1 levels go down, PGC1 alpha levels go down, MMP1 levels go back up again. The the mitochondrial cytochrome oxidase levels go back down again, and might be the rest of the components. I like the histology of the REITs. I think that might be in the supplemental. I'd have to take a look. But this is dependent on maintaining the youthful state of the mouse. So this is good that they let this experiment run its course, and adds further credence that there's factors in the young tissue, but then that actually changes over time as the mouse gets older, and the human transplanted tissue can't be maintained in that state. Okay. So now they start focusing on the actual factor that is in the title, which is VEGF-A. So let's take a look at this in figure 6. So they look at expression levels of VEGF-A RNA and also protein, because if RNA, obviously RNA is required to synthesize protein, but if you have high RNA levels, that doesn't always correlate to higher protein levels. So if VEGF-A is the actual factor and VEGF-A is a protein, and that's how it works. Some active biomolecules are obviously RNAs, but VEGF-A, vascular endothelial growth factor A, acts as a protein. So we want to test. Testing for mRNA levels is quicker and easier, but you then want to stain using antibodies for protein to show that the levels of VEGF-A actually change. And the number of human VEGF-A positive cells in human epidermis, so this is old into young, and old into old. So this is a cross-section staining within that transplantation zone. The number of these cells goes significantly up, that stain for VEGF-A messenger RNA, and the protein levels also significantly go up. I don't know if you would expect that these levels would go down again if we look at the aged mice. When the aged and the tissue ages after 12 months. I'm not sure if they did that. This is after 1 month, I believe. So this is looking at VEGF-A RNA and protein levels, xenotransplants in after 1 month. So in figure 6, they look at VEGF-A expression levels by a variety of different cell types within the transplantation zone in keratinocytes, macrophages, and platelets, both prior to transplantation, old into old, and then old into young. So let's see, how did they do this? So this is an immunohistochemistry. I don't think they isolated these cells, ran them through a fax. I believe this is immunohistochemistry and staining within the zone. So these cells that are, again, the ones that are actively proliferating in this tissue, and I believe this is expression of VEGF-A. I believe this is human VEGF-A. Got to double check this because clearly we'd like to make sure that this is actually human.
I think we don't have antibodies specific to the species. I'm not sure.
Let me take a look at this one. VEGF-A protein. You mean for the protein itself, right?
Yeah, I mean, like, we don't have specific antibodies for human VEGF-A and for mouse. We only have like VEGF-A.
Really?
I think, I'm not sure. I think this is something that they mentioned in the end also. I will look for it.
Let's see, messenger RNA. So let's see. Yeah, so for messenger RNA, they can test for human because that's probably going to be a different isoform. So for protein, you might be right about that. So they can differentiate between human and mouse with messenger RNA. For protein, maybe not. Let's take a look in the bowels of the paper here. Let's see, what figure is this? This is figure 6. Let's see if they mention that. VEGF-A protein expression. They mentioned immunohistochemistry revealed an 11-fold increase in human VEGF-A protein expression. 11-fold higher human VEGF-A protein expression.
Okay, maybe it was just neutralizing antibodies that I copied something to the chat, but
yeah, okay.
Maybe it was only talking about neutralizing. Sorry.
Yeah, that's right. It could be the neutralizing antibody experiment they do later. And they see human VEGF-A protein expression by human CD42B plus platelets. So I'm wondering if they're able to, if it's well, we could take a look at the materials and methods later. If we're really curious, they could have maybe counterstained for a human marker on a cell and then used VEGF-A that's kind of more generic. I'm not sure. That can stain both mouse and human, and then showed VEGF-A levels go up in the human cells. So they're able to at least identify the human cells within that cross section. So that's it's figure 6, and that was figure 7 I was also referring to. So VEGF-A within let's see, within these various cell types that are human within the region that they're looking at. So VEGF-A expression levels within these human cells do go up. They mentioned about roughly like 11-fold. Okay. And now this is where we sort of get into the experiments where they actually determine that VEGF-A is sort of is the key factor. It's not just, it's not just the key factor for a lack of angiogenesis not being required is actually in the tissue culture experiments, which I have to pull up in the materials and not the materials and methods, in the supplemental. So in this figure here, this is where they actually use VEGF blocking antibodies to demonstrate loss of function, that knocking down VEGF-A is actually going to have a detrimental effect. So here we have, I believe, essentially, we're doing experiments that are like figure 2, but we're adding in a different control or different experiment, which is adding in antibodies that block VEGF-A, and control antibodies, which are just non-specific antibodies. And I think, let's see, are these specific for young mice treated with VEGF blocking antibodies? And Oren, you were mentioning that, let's see, maybe this is, a non-specific, let's see if this is for against human. Let's see, figure.
There is what I wrote in the chat, like a quote there from maybe the discussion. It sounds like it is non-specific, these antibodies.
Yeah, you're right. It's expected VEGF with non-specific. That's non-specific antibodies were the negative control. VEGF-A neutralizing antibodies. Those are probably. Yeah, those are most. Yeah, they would probably have to be non-specific, because if we're, one of the models that they have is obviously VEGF-A from the mice is going to be stimulating growth in these human cells. And that's going to upregulate VEGF-A in the human cells, and it's sort of going to lead to a positive feedback loop. So for this to work, it would have to be blocking the mouse VEGF-A as well. Okay. So yeah. So we're looking now at the experiment where they loss of function, where they're knocking down VEGF-A with antibodies, and then they're looking at a variety of different outcomes, right? That are basically, so after the transplantation, they're either being injected with these anti-VEGF-A antibodies or not, and then seeing what effect blocking VEGF-A has on these different parameters. And it has the has the result that we sort of expected and that the authors expected, in that it blocks all of these effects that they've seen earlier in Figure 2 that correspond to, that are markers indicative of epidermal and dermal regeneration, right? So you have the thickness basically is blocked, so the thickness is lower. All these other factors are blocked as well. SIRT1 levels go down. Thick filament levels of collagen go down, and so on and so forth. So having these antibodies in, these blocking antibodies basically blocks these effects. And in Figure 9, they do sort of the opposite experiment, which is a gain of function, which is they have these small nanoparticles that basically are coated with VEGF-A, these PLGA nanoparticles. So not blocking VEGF-A, but basically injecting VEGF-A into the environment. And I believe this is mouse VEGF-A. I think it's mouse VEGF-A. I have to double check that. And in this experiment, it's looking at old into old transplants. So if we recall from earlier, having old into old, and the control being BSA, so bovine serum albumin, is just a protein that's used in a lot of controls, many different experiments. Sorry, as a control for the PLGA beads that are coated with VEGF-A. So these are old into old, and as we saw earlier, after kind of 1 month, and I believe this is a 1 month post-transplantation, you don't really see an effect because it's old tissue into old mice. But if you introduce the VEGF-A into basically just VEGF-A, looking at these parameters, the filaggrin levels, which correspond to the hydrophobicity barrier, proliferation, the thickness, you can see that, well, the red bar is the administration of the VEGF-A. It basically is sufficient to basically get levels back higher. And I don't know. Let's see. I'm not sure if we can do a one-to-one correspondence between these levels and an earlier figure, right? Because these are different mice. So comparing this to comparing these levels, for example, thickness 600 microns to what is it? Figure 2, which is old into young. It's about the same, almost 600 microns, the error bars there. So maybe you can do kind of a correspondence between the two sets of experiments there. I'd have to take a look at all the others. But essentially adding in the VEGF-A recapitulates a lot of these effects. There is another figure in the materials and methods, which is a shorter experiment, which I don't think we'll have much time to go into, but I'd have to find it in the supplemental. But that experiment, as I mentioned earlier, looks at the role of VEGF-A in promoting skin rejuvenation in a petri dish. It's a shorter duration; it's 6 days. It's old, I believe, old human biopsy tissue that's treated with VEGF-A, and they notice a lot of the other parameters that correspond to rejuvenated tissue also happen in tissue culture, at least in that very short window of 6 days. What's important about that is that it suggests that some of the initial positive rejuvenative effects of VEGF-A treatment is not due primarily to angiogenesis. So if you have blood vessels getting into a tissue, that was my first thought when I read this paper. When I saw the title of the paper, I said, oh, okay, well, that makes sense. You promote angiogenesis into old tissue in a young mouse, and you get a million other factors come in that might positively stimulate, growth. VEGF-A is just sort of the conduit, if you will, to basically build the plumbing into the tissue and to allow waste products to come out. So the tissue is rejuvenated, and VEGF-A is just the kind of the catalyst to start the angiogenesis. And that could be correct for kind of longer term rejuvenation. So the authors mentioned that, but it seems that the very initial effects that you see with the SIRT1 levels going up and the collagen levels changing positively, those very initial effects are not primarily due to the angiogenesis effects per se of VEGF-A, which I think is pretty cool, which suggests that it's really a very important upstream factor in promoting tissue rejuvenation, or in this case, the tissue being skin rejuvenation. So let me go back to sharing, and they mentioned this as being a main regulator. So this is all in the discussion here. And this is sort of, not as complex as this sort of looks. It's, it's always more complicated in real life, right? So this just summarizes everything that we've, and more that we've discussed so far. You have aged human skin, you have the epidermis, and you have what happens in aged human skin. A lot of, obviously, markers of senescence go up, beta-galactosidase levels go up, P16 levels go up, other things go down, like SIRT1 levels, reactive oxygen species go up, VEGF, I don't think they might be in supplemental. I think they looked at VEGF receptor as well, because the VEGF-A is a protein, it's a soluble protein that docks to the receptors on cells. So all of these things happen, all these changes. So here's the epidermis, and all these other changes happen in the dermis, right? So angiogenesis goes down, so less blood vessel formation, collagen levels go down, so on and so forth. And then when you do these experiments by adding, and they postulate here that murine VEGF-A levels in young mouse skin basically stimulates basically a feedback loop of expression of human VEGF receptor, because it docks to the receptor. You have upregulation of human VEGF-A, so you have that's kind of common in many cell types. You have a feedback loop where this now sort of positively feeds back, and you get upregulation of other factors, and many downstream effects. Angiogenesis being one of them, kind of a later downstream effect, but you have upregulation of collagen. And senescent factors going down, such as P16 and beta-gal, and so on and so forth. So I'm going to stop sharing here. So this paper, for the first time, positions at least VEGF-A as being, one critical factor that's very, very upstream of skin rejuvenation. And by blocking it, you block a lot of these effects. By adding it, as we saw here, to the mouse models that they have, as well as in their tissue culture conditions, which were, again, the caveat being that they were only a few days old in tissue culture, you can recapitulate these positive effects solely through the introduction of VEGF-A. That's not to say that that's the only factor that's going to be important downstream, but it gives strong evidence that it is a major upstream factor for all of the downstream effects that are being seen, at least in these particular experiments done with a very unique model where you can take a human transplanted tissue, in this case skin, and transplant it to the severe combined immunodeficiency mice, and then study what's taking place in the context of a rejuvenation response. So this is kind of like this is probably as close as you can get to a parabiosis study with humans, right? Kind of ethically. Parabiosis meaning connecting the vasculature between an old organism and a young organism. We do these studies in the US. I think maybe there's some countries that kind of ban it because of, animal cruelty laws. But this is kind of as close as you can kind of get as a parabiosis, I think, model to study rejuvenative effects in tissues between old and young organisms, and in this case being old tissue from humans and the host being a completely different species, a mouse. So, any questions? Any more questions?
I'm not sure we mentioned one of the experiments they did, that's transplant the young human tissue into young human skin into the old one. And then they say that the young stays young, which is pretty interesting.
Oh, yeah. Was that, I think that was in the supplement, one of the supplemental, right? It wasn't in the actual, it wasn't in one of the figures. That was in the text of the, thanks for mentioning that. Yes, that was another critical thing. In that, that suggests that, whatever effects that you don't need a continual flooding of VEGF-A, right, to promote these effects that at least externally, that if you can induce VEGF-A somehow, that it might maintain its levels. Be worth reading the paper and seeing how long they had to stimulate. I don't recall off the top of my head how long when they added in those particles, those nanoparticles that had VEGF-A. How long were they actually doing that treatment? Because what you mentioned there, Oren, suggests to me that you don't need a long-term duration of VEGF-A. It might just be a brief administration. Let's see. That would be figure 9, I think. Into young mice treated with VEGF-A loaded nanoparticles. See how long would these of course, even if they inject it once, I don't know what the longevity of these nanoparticles is. It'd be interesting to see how long the nanoparticle activity was. Let's see. It's not mentioned there. It's probably in the materials and methods.
Like in the supplementary, they say like long-term release, but okay, we can look harder. It's like in the supplementary material.
Okay. Anyway, so that's all interesting because they mentioned in the paper as well that, that again, VEGF-A is an angiogenesis factor. You don't want to be cranking these levels up and accidentally leading into tumorigenesis, right, in other tissues as well. So having a short duration, minimizing the duration of, trying to figure out what the optimal parameters are, the kind of the lowest dose and the shortest duration of VEGF-A. So if we're thinking about this as from a practical therapeutic perspective, then clearly you want to aim for the shortest duration of dosage, right, and the lowest dosage possible to attain these effects. And as they mentioned in the paper, if one of the models is the way that this is functioning is that you have a positive feedback loop, you express, that the cells themselves start to express human VEGF-A and bind to their own receptors, then that suggests to me that you don't need a continual dosing regimen to get this to work. How long those feedback loops last, I'm not sure, because this is skin. VEGF-A might be required for a number of things to basically promote skin health. But as we saw in these old long-term duration experiments with mice, it eventually goes away, right? Everything starts to age, right? So there's more fundamental changes that are happening at the kind of the basal aging level of all tissues, right? So it's not to suggest that VEGF-A is a perpetual fountain of youth, so to speak. If you're going to be adding that it might be critical to maintain organ health in the context of skin within a certain range, as assuming that all other pathways are not compromised. I think I would put it that way, right? So the VEGF-A pathways are dependent on other pathways, and if the VEGF-A level go down, then you have these problems. And if you can temporarily boost the effects higher, you maintain skin health, but all other pathways are aging as well. So if I would assume that if all the other pathways that are interconnected start to falter, then your ability to employ VEGF-A is gonna go down with time, right? So for the rejuvenative effects of VEGF-A, it'd be interesting to see how old the tissues are. Can at some, I would expect to see kind of a downward trend of effectiveness over a period of time. So that's my 2 cents on it.
Yeah, I just had a thought that we see that the young, when you transplant into the old, it's like it doesn't age. Maybe it does age, but it ages at the rate of human aging. So 1 year is nothing, 42 to 43. And maybe with the other part, it's like when we wait another 1 year in the young and old, right? So maybe then it ages at the rate of a mouse aging. I don't know.
That's an excellent thought. It's obvious now that you mention it, and it didn't occur to me. So to answer that question, we'd have to use a mouse transplant, right? So young mouse into old mouse, and see whether or not the mouse tissue ages, right? So you might see an aging effect with the mouse tissue, but not with the human tissue, because of different rate of mutagenesis happening that changes the epigenetic clocks, right? So yeah, there's a lot of things that could be still looked at. Follow-up experiments looking at the epigenetic status of these cells, looking at collagen cross-linking over time, looking at how long these VEGF effects last, in relation to skin samples that are progressively older, and also from a therapeutic experiment perspective, what's sort of the cutoff to maintain these cells in a youthful state, at least for a certain window duration, and how long. And again, tying into what I mentioned earlier, how long these VEGF-A effects, when it comes to effectiveness, actually, could last, right? Yeah. So that's food for thought, right? So again, just to kind of quickly summarize, I believe this is the first paper that does, point out VEGF-A as being one of the kind of the critical upstream determinants for rejuvenation of skin in this mouse model system that the authors have used so productively here in this paper. So I don't know what else to say.
What's this? Your last for words? Surely not. That's never happened before, guys.
Yeah, well, we try to actually, we try to wring out as much as we can. I mean, there's always much more to wring out from papers. But I think it's a really cool model too. I mean, there's kind of two cool components of this paper. They're the model system that they use to study these changes in these skid mice. And they did all the background controls to show that that for the most part the surgical technique and in these mice themselves, the background isn't really having this effect. So this could probably be applicable not just to aging in the skin, but isolating other organ systems, maybe through a transplantation model to these mice, the immune system, for example, and so on and so forth. So that's one huge, really cool component, is the system that they employ, and then of course the discovery itself, right? That comes out of using the system. So yeah, it's very good.
Good. So I hope you've all enjoyed the paper.
One quick question I have is, I know years ago I read someplace. Actually, no, it was a textbook on biotechnology by Renenberg, and they mentioned products that people have on the market, and supposedly they work. I don't want to say they definitely work. People use not VEGF-A, vascular endothelial growth factor, but they use epidermal growth factor, EGF, in certain topical creams to get rid of like wrinkles or some other skin conditions that kind of can bring the skin to a more youthful state. And I always thought to myself, how is that going to work? Because it's kind of a large protein. Can't just put it on your skin. Is there a transporter for it, or is it just, I would have to look into that, right? Because here obviously you're injecting nanoparticles, you're getting into the vasculature to get these proteins, which are not tiny. Vascular endothelial growth factor A is a whole bunch of beta pleated sheet domains. It's a homodimer, and it's not massive, but it's not a tiny protein. So just like plopping it on your skin, I would, unless you abraded the skin, I wouldn't think that you'd be able to effectively dose yourself like that. So just kind of putting that out there. I could be wrong, but I could be right.
You could be wrong or you could be right. Well, isn't that like pretty much science?
Yeah. I try to keep an open mind as much as I can until I'm proven otherwise.
Again, that's all isn't that science?
Yeah, but you would be surprised at how that's not the way it goes in science, how emotionally attached people get to certain things. And,
I wouldn't, Oliver. I know all about confirmation bias, people with agendas. They actually make the conclusion and then work backwards from there instead of the other way around. In order to sell products, there's a lot of that goes on, unfortunately, and it's done a great deal of damage to the credibility of science. I'm sad to say. I mean, you've only got to go on Facebook and look at some of the big science pages, and the amount of commenters saying, "Oh, science is bought and paid." It's a very sad state of affairs. I guess the only thing we can do is make sure that we do the best job that we can do and try to stay squeaky clean. No flies on us, right?
Yeah, that's all we can hope for. Anyway, well, thanks everybody for joining in. And thanks for the interactions. Thanks for I love bouncing ideas around. I don't know if some of the ideas that we all mentioned here, the authors might listen into what we're saying and are like, oh, that's a stupid idea, or they might be like, that's a good idea. We'll incorporate that into our experiments, or they might be like, oh, we've already thought about that. So I know. So reading a paper, it's fun to think about all the follow-up experiments that the authors or other people can do, and where all this stuff is leading. But a cool thing about this paper is that it's a really cool system to probe and test a whole bunch of other other models and to see if what other effects are happening in the context of skin rejuvenation. Is it, how much of it is through VEGF-A? What are all the other downstream components? And could we administer all kinds of other things to see if we can get VEGF-A levels to go up, right? Can this system be used as kind of a screen? So I don't know. There's the fact that VEGF-A goes up in their tissue culture system, or sorry, that VEGF-A has positive effects in their tissue culture system, is and no doubt the authors have thought about this, is basically lends credibility to automating a system, right? For having a kind of a large microplate based system with skin cells, or sort of like a little organoid system with the dermis that you can test a whole bunch of compounds and see which ones upregulate VEGF-A levels the best, right? And then administer those into the mouse system and see if those effects happen. So that's how pharma basically works, right? You try to develop a proxy system that you can automate on kind of a micro scale, and then screen through millions of things, and then take out whatever you got from there, and run it through a whole bunch of other screens, and then you have a smaller workable number of drug candidates that you can then test in animal systems, right? So the fact that and apologies that we didn't go into that supplemental, but this paper is available. So now that you've got all this background, go dive into the supplemental and take a look at their tissue-based assay. And see if it could be adopted into a microplate-based system for drug screening for VEGF-A upregulation, right? So no doubt the authors are thinking about this. No doubt they've already patented this for this process. If not, they got to hurry up. They've got a short window here. Okay. That's my free consulting. I can't hear you, Steve.
I pressed one button, but not the other. Okay, well, that was a good roundup. And you didn't go into the Captain's supplemental. Was I the only person that heard the Star Trek music when he was talking about supplementals? Maybe. I don't know.
I think you were.
I probably am. I am a bit of a shameless Trekkie. Right, so that about wraps it up. Anything else going on, Oliver, we want to talk about? Hint, hint.
Oh, yes.
No, that is so fake. Oliver, that is terrible. That was we have an amazing we've got something. It's a surprise. No, it's not. It's on our website. We have the Ending Aging Related Diseases Conference coming up, EARD 2022, for the month of August. And is it the August 11th to 14th? Am I got those dates right? It is. It's 4 action-packed days of science. It is a virtual conference, so there's no worries about traveling, fighting through the chaos that is currently the airport system.
Or wearing your pants.
Or even wearing your pants, but there is video feed. So if you're on a video feed and you're doing a presentation, please wear pants. And yes, that goes for you as well, Oliver. You're MC this year, so yeah. So
I'm MC from this point upwards.
I think he might have something to say about that. So let's keep it clean. This is a family show, guys. So we've got a big old conference, 4 days. It's I want to say it's our 5th one, because it is. So we've done it for 5 years. Each year's got, well, I think better and better. It is probably, I'm going to tentatively say that it's probably going to be the last year of virtual conference, and we're going to get probably back then to back on track like we originally were and probably host a physical event in New York. However, that said, I am pushing for a hybrid event. So for those of us for whom New York is too far or too expensive, I'm hoping that we can incorporate the virtual and the physical and create something, well, pretty special. But anyway, so August the 11th, August the 11th, July the 11th, get it right, Steve, is the conference.
No,
is it July?
Oh, August.
No, it's not. No, it is August. I was right.
Sorry, guys, you missed the conference. We all missed the conference. Everybody missed the conference.
You've all missed it. It was great. No, seriously, it's coming up on the 11th. It's coming up on the 11th, and it's going to be good. We've got all sorts of people there. We've got George Church. We've Oliver will be there as well, whoever he is. And you've got all sorts. You've got Irina Convoy. We've got Brian Kennedy as well. There's just lots, and we've added loads and loads of new speakers over the last few days. If anybody is a student as well, we are offering a very generous student discount. Get in touch with us about that. The details are on the website. And if you visit lifespan.io/conference, you can go directly there. So do get in touch if you're a student. We will be happy to help you out there. If you're a member of the press, and I actually mean a working journalist, get in touch with me as well, and we can issue press passes. We'd love to see you there if you're a member of the press. And obviously, anybody who's a hero also gets a discount. So again, get in touch. We can fit you up with a code and get you some money off. So that's it. So hopefully, we will see some of you there. We've got a fantastic lineup, and of course, if you do attend and get a ticket, the event is actually accessible for at least a month afterwards. So if you can't necessarily watch every single talk because you're doing some other things, you will have the option for at least a month to be able to log in and replay it on demand. So that's the big news that's coming up. So enough of that now. So if I haven't convinced you now, I'm not going to convince you by another 5 minutes talking about it. So I will either see you there or I won't see you there. Oliver, I'm hoping you'll be there.
I hope so too.
And I hope you're going to have no more of your problems. So remember, if you do have any strange things going on, if you do have some strange things going on, reboot and try again. That's the golden rule with IT.
I will cut the power cable and splice it back. No, I won't do that. I'll reboot and try. Yeah.
Always reboot. If your computer's acting weird, try a reboot. It fixes 99.9% of sins. Because computers get a bit mixed up sometimes. Right.
All right.
Yes, we will all see you next time. We don't know what the paper will be. I'm sure Oliver will find something exciting.
Yeah, and I keep asking for requests, and we do have this paper. Oh, by the way, this paper here, if you want to read a summarized blog article, we do have this paper on our science blog on lifespan.io. So do check it out.
Maybe we'll look at that vitamin D thing, Oliver. There's been quite, shall we say, a lot of heated debate about vitamin D and Alzheimer's. I'm sure you. In fact, I know you've seen it because you've commented about it in the chat room that we use. It is pretty controversial, but somebody is basically bringing under fire a popular hypothesis for Alzheimer's and vitamin D, I think it was. But there seems to be some problems with the study because it's not. I understand it's not exactly vitamin D that they use. They use a derivative or something. But there's been a right old, very heated debate, shall we say, over that. So that might be interesting to dabble in a little bit of controversy. See if we can reach any conclusions.
Yeah,
maybe.
We'll see.
Just putting it out.
Yeah. Again, this field is going really fast. Papers are churning out left, right, and center. So, the problem is that, I see a paper I like, and then as Journal Club approaches, something else just comes out of nowhere, and it's like, whoa, that's really cool. Let's do this, right? So, that's a good thing. lots of papers to cover, right? And hard to basically kind of tread water, but we try our best. So, yeah.
Right. So we'll see you next time, maybe with something slightly controversial and spicy. Or we might just find something else. I don't know. But we'll let you know the usual time, the usual places. We will be in touch, as they say. So,
all right, everybody.
Take care, everybody. We will maybe see you on the 11th of one of the months.
All right.
See you, everybody.